Hi everyone, welcome back to the Viking Initiative for Building Excellence. This is Abibayo, and today we're going to talk about water potential. This is an additional higher level topic, so if you are taking SL, then you do not need to know about this topic. Okay, so water potential, all it is the measure of the potential energy of water. It is contingent on two factors. So one of those is hydrostatic pressure. It is directly proportional to this, and the other is solute concentration. It is inversely proportional to this, meaning that if hydrostatic pressure increases or decreases, water potential will do the same. It will do the opposite of solute concentration, depending on whether it increases or decreases. Um, this is the measure relative to water at sea level, where the water potential is zero. The units of water potential are either kilopascals, megapascals, which is just larger, or bars, which is what you'll see most of the time. Okay, so if we're talking about water movement in terms of water potential, so we know that osmosis occurs when solid particles are too large to diffuse on their own. So water has to move in the opposite way to balance out the solute concentration. So it will move from a area of high water potential to low water potential. And we know that hydrostatic pressure is proportional, directly proportional. So that means it'll move from an area of high hydrostatic pressure to low hydrostatic pressure. And we know that it is inversely proportional to solute concentration. So it will move from an area of low solute concentration to high solute concentration. This makes sense because if the concentration is low in one area and high in the other area, water will move to where it is high in order to balance out the concentration. Okay, so now we're going to look at the different components. So let's replace solute concentration with solute potential and hydrostatic pressure with pressure potential. So solute potential is always negative, and it becomes more negative with the addition of solute into water. So it is zero when there is no solute, and it becomes negative with the addition of solute. Pressure potential can be negative or positive relative to when the pressure is zero. In this sense, water potential equals the solute potential plus the pressure potential. Now, sometimes you will have to calculate solute potential. This may be with a graph or in sort of a database question. So there are two constants in this equation, the ionization constant and the pressure constant, both of which will be given to you. And notice that in this, the units for pressure constant is liters times bars divided by moles times Kelvin. So here, the units for solute potential will be in bars. Um, the C stands for molar concentration, and you would just determine this from your experimental data or the experimental data given to you. And lastly, T is the temperature in degrees Kelvin. This is very important. They will often give you degrees in Celsius. You need to add 273 to convert it to Kelvin. Okay, so let's look at the application of water potential in plant and animal cells. So there's two main conditions. First is hypertonic. So hypertonic means that the solute concentration of the environment is higher than the concentration in the cell. So we said that it moves from low concentration to higher concentration, which means that water rushes out of the cell into the environment where the concentration is higher. This means that because there's less water now in the cell, the pressure potential decreases. And when the pressure potential decreases, we know that the water potential decreases. So when it's in hypertonic solution, the water potential decreases. You can see in the picture, in an animal cell, in hypertonic solution, it becomes shriveled. In a plant cell, it becomes plasmolized. Two different words for this, but the same concept. In hypertonic solution, it is the opposite. The solute concentration of the environment is lower than in the cell, so water moves where it is higher, so it rushes into the cell, increasing the pressure of water against the walls of the cell, therefore increasing the pressure potential and the water potential. So you can see in a hypertonic environment, the red blood cell becomes lysed, and in the plant cell, it becomes turgid. So isotonic solution is where it wants to be. This is the reason for water movement as a whole. It is moving so that solute concentration and pressure can reach equilibrium. In a red blood cell, it'll look normal, and in a plant cell, it'll be flaccid, right? So it won't be so bulged out, but it also won't be shriveled. Okay, so let's do some practice MCQs, FRQs, and DBQs. First, a root vegetable is placed into 5 sucrose solutions of different concentrations 0, 0.1, 0.2, 0.3, and 0.4. Its osmolarity is 0.25, and how many solutions will it lose mass? Osmolarity sounds like a complicated word, but this is the concentration at which the vegetable is at equilibrium. This is its isotonic concentration. So you know it will lose mass when water rushes out, meaning that it is hypertonic, right? Water rushes out to a higher concentration outside of the cell, so it'll be in hypertonic solution. And which of these is hypertonic to 0.25? It'll be 0.3 and 0.4. So therefore, in two of the solutions, it'll lose mass. Number two, a red blood cell is placed into a 0.5 mole solution and it becomes lysed. Which of the following could be its osmolarity? So we know that when it becomes lysed, if we go back a slide, it means it is in a hypertonic solution. Lysed means it bursts. So its osmolarity would have to be greater than the solution that it is in because the solution is hypotonic to the red blood cell. The only one of these that is greater than 0.5 is 0.6. Okay, now on to FRQs. A student calculates the solute potential to be negative 2.0775 bars, given that I equals 2, C equals 0.5, R equals 0.0831, and T equals 25 Celsius. What is their mistake? If you take the time and do the math, you multiply 2 by 0.5 by 0.0831 by 25, you will indeed get negative 2.0775. But as I said before, they forgot to convert the temperature into Kelvin, so this is wrong. Number two, water potential has a blank relationship with solute potential. We know that it is inverse. As one increases, the other decreases, and vice versa. Lastly, one database question. This graph illustrates the change of a potato's mass when placed into sucrose solutions of various concentrations. So first of all, what is the osmolarity? We know that this is when it is isotonic and when the potato has not lost any mass. So you want to find where the change of mass is 0, and we can estimate that the osmolarity is around 0.36 or maybe 7 molar units, okay? And part B, at what concentration would the potato gain mass? So it would gain mass when water rushes in. It would rush in because the concentration of the potato is higher than the concentration outside the potato. So this means that the concentration would have to be lower than its osmolarity. So any point left of where this line hits the 0% line is where it would gain mass. All right, that's it on water potential. A pretty small topic, but can be a bit confusing. And the FRQs on this topic can definitely be a bit challenging. The one like that potato is a very common type of structure for an FRQ on this, so look out for that. As always, visit the Viking Initiative website for more resources. Feel free to join the group chat and ask us if you have any questions. And thank you for watching.